All right. Okay, right. Okay, we should be now presenting to everybody. This is, can everybody see that? Frontiers in Molecular Bio.
Yes.
Kudoke?
Yes.
All right, I'm going to expand that. I think I'm going to expand that. There we go. Everybody sees that. Great. Okay, this is the last lecture I'm going to give on the view from the top of the mountain, from what you can see if you've got a working knowledge of DNA, transcription, translation, and all of that. And starting next week, we start going down. I'll be able to talk more about cells, and we'll end by talking a bit about ecology. But this is the last lecture where I'm really going to hammer on DNA and all that. Yuki, if you could mute your microphone a bit. I'm getting some noise from there.
Okay.
Okay, sorry about that.
No, you're fine. I'm sorry.
Okay, right. You already know one great scientist who got his PhD from the University of California at Berkeley. Here's the other one. Actually, there's been more than two, but the first is, of course, me. And the second is Dr. Carey Mullis, biochemist and also a surfer dude who was working for a San Francisco biotech company that I don't think exists anymore, or I think it's merged with a bunch of others, but it was called Cetus back in the 1980s. And so he was working on biochemistry and surfing a lot and taking a lot of drugs. And in 1985, he got the idea for a technique that would take any little snippet of DNA, any piece, and copying it exactly millions and millions of times over. You could take one little piece of DNA and copy it as many times as you cared to. And yes, he got some of his idea after taking some LSD. I don't recommend this as a technique for scientific discovery. Remember that hypotheses are very creative acts, and sometimes scientists have used recreational chemistry to come up with creative hypotheses, although I'm not actually going to tell anybody to try that. There were some others who had come up with some similar ideas, and Mullis wasn't working alone. Scientists really don't do that anymore. We work in teams. But he was the prime mover in getting the idea to work, and he won the Nobel Prize in chemistry in 1993, and then wrote this kind of weird book called Dancing Naked in the Mind Field, which I'm not actually going to recommend. The guy is a fascinating person, but dang. Anyway, what did he come up with? It's called the polymerase chain reaction, or for short, PCR. And it involves a lot of those little plastic test tubes. I don't remember if you guys got to this lab or not. Did you do the lab that involved gel electrophoresis? Somebody real quick, unmute your mic. Did you guys get to this lab?
No.
Okay, I think you poured the gels for it, right?
No.
Okay. All right. Well, this is a lab that you would have done, and unfortunately, I guess we just won't be able to. But those little plastic vials are used in PCR for holding DNA samples. Now, you might remember a long time ago, I mentioned extremophiles are organisms that can live in environments that are too extreme for anything else, that are too hot or too cold, too salty, too acid, too heavy metal. environments where nothing else can. And you might remember I mentioned that a lot of this group called the archaea are extremophiles, although not all of them are. The picture that you're seeing is a bacterium called Thermus aquaticus, and it's not an archaean, it's a bacterium. It's in that domain bacteria, nice long bacillus right there, and it happens to live in hot springs. It happens to live perfectly happily at, I believe, about 55 degrees Celsius, something like 150 Fahrenheit. And like all living things, it replicates its DNA and it uses enzymes to do the job. There are little proteins that move down a strand of DNA and unzip it and bring in new nucleotides and assemble new strands. For most organisms, if you heat them to that kind of high temperature, the enzyme will denature and it won't work. If we heated you to 50 degrees Celsius, your enzymes would all start to denature and they'd shut down and you'd be dead. It doesn't even take that much. You start running into trouble at, with fevers of about 104 Fahrenheit or maybe 41, 42 degrees Celsius. But Thermus aquaticus can do it just fine, and they've got a very stable DNA polymerase. So what you can do is take any strand of DNA, heat it, and if you heat it a bit, the hydrogen bonds will break and the DNA will naturally unzip. And then you hit it with DNA polymerase, and that makes new strands on each of the old. And then you heat it again and hit it with polymerase, and heat it again and heat it with polymerase. You cycle it over and over again, and you end up with 2, 4, 8, 16, 32, 64, ... millions of copies of any piece of DNA that you fed into it. So there on that slide, I basically just explained that. If you heat DNA gently, not enough to burn it, you unzip it, you break those hydrogen bonds. That DNA polymerase, which is shown in a diagram on the left, builds new strands, and then you cool it down, and then you heat it again. You're doing all this in a basically a metal block with holes in it called a thermal cycler that heats and cools on a very precise time frame. Repeat this over and over, and you have DNA copied by the millions. We don't really have time to get into exactly how you modify this, but you can modify it in a nifty little way to make it possible to sequence DNA directly. You can take one lousy piece of DNA, copy it millions of times, use this little addition to the technique, do this thing called gel electrophoresis, which I'll talk about in a minute, and read the order of nucleotides off. If you look at the top of that picture, you'll see that going from left to right, you have 4 lanes that are labeled T, G, C, and A. You just read those from left to right, almost like reading notes on a staff. So at the top from left to right, that says G, G, C, T, T, C, G, A, A, G, 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 A, C, T, A, C, A, and so on. And yes, for the music majors here, somebody has experimented with taking DNA sequences and turning them into sequences of musical notes. Since there's only 4 notes, it's not all that interesting, but it's the kind of thing Philip Glass would probably really, really like. Anyhow, moving along. And it's PCR that makes it possible to identify DNA samples from individual people. This is the kind of thing that they're always doing on shows like CSI and Law and Order and things like that. Although in real life, it can be harder than that, and it usually takes longer. You usually can't get it done within the confines of a 30-minute TV show. But it does work, and it's used to solve crimes. And there's a number of different ways you can analyze DNA from a crime scene and try and trace it to the person who might have left it there. We don't have time to get into all of them. In general, they're called DNA fingerprinting. And I'll talk about one in particular that's in use by the FBI. And it looks like this. The DNA that makes up your chromosomes carries all your genes, but there are long parts of your chromosomes that are made of DNA, but they're not genes. They're not transcribed and translated to make any protein. Some of them are involved in just holding the thing together. Some are involved in control of the transcription process. Some of them don't seem to be doing anything. And one of these that doesn't seem to be doing anything are sections that are called short tandem repeats, STRs, if you like. And an STR is just a stretch of your chromosome where the DNA just repeats a short sequence over and over and over again. CATG, 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 da 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 da. For the music majors here, this is kind of the equivalent of Hanon piano exercises, if you ever had to do those. STRs vary in length from person to person. Usually, I can think of exceptions, but usually it doesn't make any difference. I might have a spot on one of my chromosome 2s with 7 repeats of CATG. Nicolette, you might have a spot on your same chromosome that has 9 repeats, and Aaron might have 15, and Yuki might have 12, and so on. People have different numbers of these short repeats, and it doesn't make any difference to your life or how you function, at least not most of the time. They're just different from person to person, and they tend to shift a lot because of mistakes in the way chromosomes pair up in meiosis. What you need to know is that people have different numbers of repeats in their STR sections. And so you can copy a DNA from a crime sample, run it through a process called gel electrophoresis that I'll show you in a minute, even though we couldn't do it in lab. You can measure the length of DNA fragments and then compare the STR lengths in an unknown sample with the STR lengths in a known sample and see if they match. This is what you guys would have done. In gel electrophoresis, you take samples of DNA fragments and you put them into these tiny rectangular holes called wells in a gel. And a gel feels like jello or gelatin. It's actually made of something else. It's a carbohydrate called agarose, but it feels very soft and floppy, and it's clear. And DNA can actually move through an agarose gel. And then you put the gel in a little plastic frame and you apply an electric current to the gel. You put a positive electric charge on one end of the gel and a negative charge on the other. The reason that matters is that DNA has a slight negative electrical charge, and it's attracted to the positive side of the electrical field. It's attracted to the positive electrode. And fragments of DNA will move through the gel. They can do that, but smaller fragments can pass through more quickly than large fragments can. A bit like why, imagine you had a very tangled forest with lots of vines and shrubs and no clear place to run. And imagine you and a mouse were running a race. The mouse, being a lot smaller, can go through all of that tangled stuff. You, being bigger, are going to take a lot longer to push your way through. It's the same for DNA moving through an agarose gel, which on the molecular level does actually look kind of like a tangle of underbrush and shrubs and vines and all of that. So the upshot is that if you put DNA fragments in one of those wells and turn on the power, you sort them by size. The smaller pieces of DNA move farther in a given length of time than large pieces of DNA do. That's how gel electrophoresis works. And you can measure the size of a piece of DNA very quickly and very accurately. All right. Everybody having a good time so far? Okay. All right. I'll take that as a yes. Okay. For any single one of these STRs, 5 to 20% of the human population will have the same length. One of my STRs might have, I don't know, 13 repeats of something, and maybe Kayla's will also have 13, and there may not be any interesting reason for it. Now, you inherit these like you inherit all of your DNA from your parents. So if I have 13, my dad might have 13 as well, or possibly my mom. Kayla and I could be related, but it could also be just sheer coincidence. We might have the same number, but it may not be for any reason other than that's just how it worked out. But the more STRs you use, the lower the odds that two people will have exactly the same length at all of these STRs. If I've got 4 STRs that are 13, 7, 6, and 8, it's much less likely that Kayla's going to match all those 4. Until fairly recently, the FBI, the Federal Bureau of Investigation, maintained a database called CODIS, the Combined DNA Index System. And whenever they took DNA from a crime scene, or in some cases from a criminal, what they would do is measure the size of 13 different STRs that were called the core 13. More recently, they've upped that to 20. And the probability of two people who are not related having all 13 STRs happen to match up is 1 in 1 quintillion. That's a 1 followed by 15 zeros, which is, think Wagner, 9, 10. That's what, 10,000, more than 10,000 times the population of the entire Earth. So the odds that two people have the same 13 core STRs is astronomically low. And that's the data that they gather and put into databases when they're trying to solve crimes on CSI or whatever it is that you watch. This is just a diagram of where those core 13 are. You see diagrams of human chromosomes, 1 through 12 at the top, and then 13 through the sex chromosomes at the bottom. You might remember that chromosomes show banding patterns when you look under the microscope, and you can see those. And then each of those yellow tags is the location of one of the STRs. You certainly don't have to know the codes. This is just what they're called. So on chromosome 2, you have an area of your DNA that's called TPOX, so called because it had a feud with Biggie Smalls. I'm sorry, I'm kidding. Never mind why it's called that. It's just a point on one arm of your chromosome 2 where you have one of these STRs, where I might have 12 repeats in my TPOX and somebody else might have 7 or whatever it is. And then on chromosome 3, there's an STR called D3S1358, so those are the 13 that are used by CODIS. These days, they've expanded it to 20. They're actually looking for, there's some more that they can use on sex chromosomes. They're expanding this all the time. But this is the basics of it. And that's what it looks like. That's a DNA fingerprint. Up at the top of that diagram, you see a bar that's labeled D19S433. That's just the name for an STR. It's a part of chromosome number 19, and it's got an STR on it. And one copy of this person's chromosome 19 has a 13 repeat D19S443 STR, and the other copy of his chromosome 19 has a 14 copy D19S433 STR. And then down the line, VWA is another STR, and it happens to be on chromosome 12. And one of this guy's chromosomes 12 has a 14 repeat VWA, and the other copy has a 17 repeat VWA. And then at the right, TPOX is on chromosome 2. He's got 2 copies of chromosome 2, and both of them have a TPOX STR that's got 11 repeats, and so on. So that's the data that they're looking for. And I hear a beep, so I'm going to pause this for a second and check in on, okay, Jonathan Blair is joining our ranks. Welcome. Hi there. How's everything? Okay, I can see you nod. John, can you see me? Mr. Blair? Okay, I'm seeing you nodding. Great. All right. Going back to the slides, we've been talking about how DNA is used in forensics, in tracking criminals. And if you missed the first part, we'll be uploading this to YouTube and to Blackboard, so you'll be able to watch the parts that you've missed. Right. Okay.
My fault.
Sorry?
I said my fault. My stuff was muted.
Okay. That's fine. Okay. Going back to where we were. All right. Can everybody see that? Whoops. Okay. Real. Okay. Go back and watch the part that you missed. There are some worries about privacy issues there, because there's certainly ways you can misuse DNA evidence. There's cases where the government wants to collect it when they don't have an obvious reason to. This is one of the things that they test your DNA for if you go to one of those places like 23andMe, and those databases aren't necessarily private. So there have been cases of criminals getting caught when they or their relatives submitted DNA samples to companies like 23andMe, which is probably a privacy violation. But there are cases where it's been used to overturn some wrongful convictions. The gentlemen in the center of the picture are Levon Brooks and Kennedy Brewer. They were imprisoned in the early 90s, both for raping and murdering children. They maintained their innocence all this time. And when word of their case got to a group called the Innocence Project, that's a legal charity that successfully petitioned to have some DNA testing done on the forensic evidence that hadn't been done. I'm not sure if it was doable back when the crime was committed. Both of them were found innocent, and the actual perpetrator of both crimes confessed. So the Innocence Project reports that as of March 2020 in the United States, 367 prisoners, including 21 who had done time on death row, have been exonerated by DNA testing. So there are certainly very positive ways in which this can, in which the technology can be used. My personal view is that it's neutral, but there require controls to make sure that it's used in positive ways and not potentially in negative ways. You are free to have any opinion on the matter that you want, but it's good to see people going free and getting at least some of their life back from wrongful imprisonment. Right, carrying on. Different groups of people all over the world have different sets of STRs. Normally, these don't really have any particular effect on gene function. They don't do anything. They don't make any real difference to the way that you live your life. But this is neat because you can use these to trace human ancestry. What you're looking at is a map of a set of STRs on the Y chromosome. That set of STRs is collectively called haplogroup Q. It's just a name. It's just called group Q. And it's a name for a set of STRs and single nucleotide variations on the Y chromosome. And this particular set of STRs is rare in most of the world, but it happens to be very common in Native Americans in both North and South America. All of that darkly purple shaded region is areas where between 60 and 100% of people have it. This, by the way, is Native American populations. You're seeing immigrants from Europe or descendants of Africans don't have a high incidence of haplogroup Q. You'll notice that there happens to be a spot in the middle of Siberia over on the left that also has an unusually high incidence of haplogroup Q. It's that area that looks like a purple bullseye. What that suggests is that there is a genetic link between people in that part of Siberia and the population of the natives of North and South America. Now, we already thought that Native Americans are descended from Asians who came across the Bering Strait from Siberia into Alaska. And here it looks like we've pinpointed either a place where the ancestors of Native Americans come from, or some people could think it was some Native Americans who traveled back. By itself, you can't tell which of those is true, but you do have a definite genetic similarity between some populations in Northern Asia and the Native Americans. And it's things like this that there are African Americans who are using this to try to identify the parts of Africa where they came from. Most African Americans are descended from slaves, and nobody bothered to keep any records of where in Africa they came from. So some African Americans feel like they don't really know what their family roots are. And this is one way that you can get some information, not everything you might want to know, but at least a bit on where your roots are located. My dad actually had this done. He's got STRs on his Y chromosome that's called group R1A, which basically means he's Northern and Western European, and we already kind of knew that. I mean, my family is basically, makes sour cream look tan. But yeah, people of all backgrounds can sometimes use this as a tool. It also provides whole new ways to reconstruct evolutionary history. You can download DNA sequence data from a staggering number of different organisms whose DNA has been extracted and sequenced. The site is actually called GenBank, and it's maintained in the US by a government agency called the National Center for Biotechnology Information. There's also a European center and a Japanese center that store genetic information like this. And as a tiny sample, you're seeing snippets of actual DNA from, at the top, a hippo, and below it, 3 different whales. The orca or killer whale, the sperm whale, which also has teeth, and the blue whale, the largest animal of all. And these are all pieces of DNA from a gene. I believe this is called cytochrome B. Don't worry about what it does. And first of all, you can see a lot of the nucleotides match up. They all begin with T. The 3rd and 4th nucleotides are C for all 4 of these. They all end in G, and so on. You can see a lot of similar, of identical nucleotides between the 4. But what the red boxes are showing you are places that are different. So the 2nd position, the hippo has G, the orca has G, but sperm and blue whales both have A. And then in the 5th position, hippo has C, but orca, sperm, and blue all have A. And then in the 6th position, hippo and the blue whale have a G, but the orca and the sperm whale have an A, and so on. I'm not going to read the whole thing. What those result from is past mutations. Somewhere down the line, some ancestor of the sperm and the blue whale underwent a mutation that changed the G in the 2nd position to an A, and so on. Am I making sense? I trust I am. And doing this by hand is one of the biggest pains in the neck I've ever had to do, but that's why the gods gave us computers. So using computer analysis methods on very large sets of DNA data, much bigger than what I just showed you in the past slide, we can look for similarities and differences in nucleotides to draw these things called cladograms, which you might remember from a much earlier lecture, these diagrams of common ancestry. So that is one based on DNA data. Don't worry about the little letter and number codes like INOARE2 and things like that. Those refer to DNA sequences called short interspersed nuclear elements. Don't worry about those. We don't need to worry about them here. But you can see that toothed whales and baleen whales are down there at the bottom. They share a common ancestor, we think, based on the pattern of similarities and differences in DNA with hippos. And then whales plus hippos share a common ancestry, we think, because of similarities and differences in DNA with chevretains, which are little antelope-like things, and pecorans, which is everything from deer and giraffes to cows, sheep, and goats, and antelope, things like that. And then all of these share an even more distant common ancestor with pigs and peccaries. And then those share an even more distant common ancestor with camels, all the way back to here. That is a cladogram that's so big we've had to bend it into a circle. And it's based on a very, very large DNA data set. And up there at the top left, you can see it says metazoa, right next to an enormous number of branches that take up about half the tree. Metazoas, for our purposes, a fancy word for animals. We are located as one little branch on that tree, somewhere around where it says you are here. Somewhere in there is us. But fungi make up a big branch down there. You can see archaea at the far right, bacteria at the right. Those other names are different groups of protists. Remember, I said that most people divide up the kingdom Protista into different kingdoms, and we don't really have time to explain why. Those are some of the groupings that we use. And we can use data like this to explore the evolution of all life on Earth from the last common ancestor of it all, which lived approximately 4 billion years ago, from archaea through bacteria, eukaryotes, all the way to us, from the goo to the zoo to you, as we say. And we can even use this to track the evolution of virus stains. You probably don't really want to hear about COVID-19. You're probably quite sick of it. Well, I hope you're not literally sick of it. I hope you're just tired of it. But what you're looking at is a cladogram based on sequences of DNA, and every one of those names is a different strain of virus. The blue arrow kind of in the middle is pointing to, it says human MERS-CoV, which is very closely related to something that says camel MERS-CoV. That's in the center. MERS is Middle Eastern Respiratory Syndrome. That was a health crisis in the Middle East a few years ago, although I don't believe it made it to the United States. Down there below in red boldface, Beta CoV Wuhan, all of those branches, that is your friend and mine, coronavirus 19. And you can see that its closest relatives are, in fact, viruses that live in bats. Viruses can jump from humans to non-human animals and back. China has gotten some criticism for this because of their use of those wet markets, but it's happened in other cases as well. HIV is most closely related to viruses in chimpanzees, and that's thought to have made the jump possibly as early as the 1950s. Hantaviruses sometimes make the jump from rodents to people. Just one of those things that viruses do. And the ultimate reason why that viruses keep changing is that as they replicate, they mutate, they make errors in DNA replication, or RNA sometimes. Sometimes those errors mean the virus doesn't do a very good job of reproducing. Sometimes those errors improve the virus's ability to reproduce itself. Sometimes those errors make the virus more efficient at getting to hosts and infecting them. So the reason why all of this evolution is happening is our old friend mutation plus selection. It's good old Darwinian evolution back again. This is the reason why you keep needing new flu shots every year, is that the influenza viruses are constantly mutating, and most of those mutations don't make for a more dangerous virus, but every so often one pops up that is new and does pose a hazard, and then we have to develop a vaccine against that. That's why you have to get a flu shot every year. And we can track this process and see down to the nucleotide level where and how this is happening with this DNA sequencing that we can do, this polymerase chain reaction that makes things like this possible. So, yeah, probably you don't want to be reminded of COVID goddamn 19, but at least you can get some insight in some of the tools that we're using to try to understand it and hopefully try to fight it. A lot of smart people are working overtime right now doing exactly that. I'm going to pause for a second and cut over here. Any questions? Looks like some people dropped out. I'm seeing about 6.
Hey, I got a question.
You go right ahead.
This Jonathan,
I see.
this Blair.
Yeah, I see you.
All right. How do you do the lab assignments just do? And like that's due on the 3rd and the 10th.
Okay. Well, you'll need your lab manual.
My lab man. All right.
All right, and we've uploaded to the department website files that you need in order to fill those out.
You said what?
We've uploaded to the department website some files that you need to finish those labs, and I emailed out the link. I'll do it again this afternoon.
So do I go to the exploring concepts in biology?
Oh no, it's not the it's not the textbook. It's department. It's a link at www.uca.edu/biology. I'll send it to you after this lecture.
Okay, bet.
All right. Okay. All right.
Oh, one more question.
Sir.
We haven't had any work due since like this corona stuff, or we have?
Do we have to have work due?
Have we had any work due?
Other than the labs, no. I'm trying to come up with a quiz that I can give you guys remotely. But aside from the labs, there's no pending assignments just yet.
So the only assignments that's been due is the one that's due on the 3rd.
We've got the next lab, number 7, is due on the 10th, and they're working on getting data for the lab after that ready.
No, I'm saying before the one on the 3rd. Was there ones previous assignments or no?
No, there was not.
All right.
All right. Okay, let's finish this. Okay.
I'm sorry.
Yes, Yuki.
I'm sorry. In addition, in the like upcoming Friday, due on Friday,
Yeah.
there's nothing. The like assignment, like lab assignment, like I guess the chapter 7 lab assignment is upcoming
Right.
Friday. So, but you said it's on 10th. So,
There's one lab 8 is due on the 3rd,
yeah.
and lab 7 is due on the 10th. And
Okay. I'm sorry. Okay, thank you. Yeah.
All right. I will send out an email reminder.
I'm sorry. Thank you.
All right. Quite all right. Okay. Right. Ultimately, this finally answers the question that Darwin couldn't solve, which is how do you get variation for natural selection to work on? You might remember that we left that hanging at the end of our unit on evolution. The ultimate way that you get new alleles is mutation. It can be as simple as changing one nucleotide. It can be as complex as duplicating entire genes, but that's ultimately the way you get new alleles. We can sometimes see this happening. You might remember a long time ago, I mentioned that cat with curly ears. That was an example of a mutation that gave rise to new alleles and a new trait which breeders could select for. And now we have a whole breed of curly-eared kitty cats. We can measure how common alleles of a gene are in a population, and I think you guys did do that. This was the lab with the brown and the white beans. Did you guys do that one? I thought we were setting up for it.
We did.
You did?
Yes.
Okay. I don't know, maybe the Friday lab might not have, but
Yeah, no, I'm in Friday lab and I don't remember us doing that.
okay, well, Friday lab got dismissed that week. I think this would have been the very last lab before break.
Okay.
So about 2/3 of you have done this lab and the other third hasn't, I'm afraid. But it was simulating how you can calculate the frequency of different alleles of a gene are in a population. That's called the allele frequencies. And allele frequencies can change from one generation to the next, and sometimes it's due to natural selection. Some alleles become more common, other alleles become more rare. Sometimes this is selection, sometimes this is due to entirely random effects called genetic drift. But let's look at a case study of this before I let you folks go. That's a European moth called Biston betullaria. The common name is the peppered moth. These are the same species. They can interbreed perfectly well. The difference is essentially one gene with a few alleles. One genotype gives the moth dark wings. Another genotype gives the moth these light speckled wings that you see at the top. And dark happens to be dominant to light. There's a few more alleles for kind of intermediates in between. We don't really need to worry about those for our purposes here. But yeah, this is the same species of moth. It's just some have, you know, at least one allele for dark colored wings, and others are homozygous for an allele of the same gene for light colored wings. This follows good old Mendel's rules. Now, in England, there are some collections of moths that date back centuries, and light moths were far and away the most common before about 1850 in England. Beginning about then, light moths were almost completely replaced by dark moths. Dark moths became far and away the most common. Light moths became much more rare. But since 1959, they have been at experimental plots in England. They've been collecting and counting moths every year. And you can see that x-axis on the horizontal plane begins in 1959. And the percentage of dark moths is what you see on the y-axis. And in 1959, 95% of the moths were dark. You can see how that began declining, first slowly, and then beginning about 1975, it starts declining more steeply. And as of 1995, about 85% of the moths were light, and only 15% of the moths were dark. And it's not just dark moths, it's the alleles that make them dark. Those alleles have become much more rare in the British moth population than they used to be. And the same thing was observed in the United States. The triangles that you see down at the bottom of this graph is the frequency of dark-colored moths in rural Virginia. Virginia has some cities, but there's large areas that are still very underdeveloped, very beautiful, mountains and forests and things like that. Great place to visit. And the peppered moths there have always been mostly light. There've never been many dark-colored moths, fewer than 5%. The circles show you moth populations in Michigan and in Pennsylvania. Those are industrialized areas. And you can see that most moths were dark in 1959. By 2001, fewer than 10% of the moths were dark. So we have the same kind of change at the same time in the eastern United States, in Britain. It's also been observed in Germany and Japan. And the reason is this: light-colored moths blend in very nicely with the light speckled bark of trees that are covered in lichen. If you don't know what lichen is that light-colored crusty stuff that you can see on tree bark. There's lots of it on campus. There's probably some where you are as well. There is a light-colored moth down at the lower left of that picture, but it's very hard to pick it out, whereas that dark-colored moth is very easy to see at the right and a little bit above. So everybody can see the dark moth, but that light moth is well nigh invisible. The problem is that in industrial areas with lots of pollution, soot covers exposed objects, including trees. You may not have seen this, but areas downwind from factories and things like that used to get completely covered in dark soot on a regular basis, up until the passage of clean air legislation in the 50s and 60s. Lichens tend to die in polluted areas because they happen to be pretty pollution sensitive. They'll get killed off. Trees with light-colored bark also tend to get killed off by pollution, notably birch trees. And the upshot is if there's no pollution, most of the places for moths to live on are light-colored, and the light-colored moths get hidden very well. They're camouflaged, and the dark-colored moths get eaten by predators because they're easy to spot. On the other hand, when the backgrounds are dark, light-colored moths get eaten by predators. The dark-colored moths are likelier to survive because they blend in. And you probably have never seen pollution like this, but this is what downtown Pittsburgh looked like in the 1940s. It's one of our big industrial cities of what's called the Rust Belt. Pittsburgh is famous for its steel mills, for glass, for paint, for all kinds of things. And before the adoption of pollution controls, all of the soot and chemicals from heavy industrial manufacturing was just released right into the air. It made Pittsburgh an economic powerhouse, but Pittsburgh also used to be called hell with the lid off. The pollution was so bad that they used to have to light lamps in daytime. London used to be like this. Big cities like, Allentown, Pennsylvania, and Bethlehem, Pennsylvania, and Chicago and Cleveland, all of these big industrial cities in the Northeast United States used to have pollution problems like this. And I'm pretty sure none of you have ever had to live with this sort of thing because far as I know, everybody here has lived all of their lives under laws that restrict the amount of pollution that you can release. There are air scrubbers and filters and things that reduce the amount of pollution coming out. But between about 1850 and 1950, there weren't any controls on this sort of thing. And I've seen old neighborhoods in Pittsburgh where the buildings have turned completely dark for no reason other than huge amounts of soot that has built up and is still there on the downtown buildings. And it has the same effect on trees, or it used to. So pollution like that would have had a huge effect on peppered moth populations in the area, and alleles for different wing patterns in peppered moths have changed in frequency accordingly. Dark became very common when backgrounds were dark because of pollution. Light has become more common now that the pollution has slacked off and the backgrounds that the moths live on has become lighter. All right, did that make reasonable sense? All right, any quick questions?
I'm good.
Okay, all right, I'm going to stop recording now. Okay, take that back. I'm going to stop recording now.